Peroxisome structure. Peroxisomes are single phospholipid membrane-bound organelles. Here we can see the bilayer of the peroxisome membrane. They are an incredibly dynamic organelle and they are found in almost all eukaryotic cells. The size varies from 0.1 to 1 micrometer in diameter. In certain tissues, such as in the liver, they are larger, being up to 1 micrometer. In others, such as fibroblasts, they can be as small as 0.1 micrometers in diameter. The average dry weight of a peroxisome is 24 times 10 to the minus 15 grams. Peroxisomes are usually ellipsoid or spherical in shape. There is random sectioning and compartmentalization within the peroxisomes. Hourglass-shaped reticulum bridges may or may not also be present. These bridges connect peroxisomes. The peroxisome may also be in close association with smooth endoplasmic reticulum, mitochondria, or chloroplasts. There is an inner matrix which contains many enzymes relative to function. They can contain up to around 50 different types of enzyme which are involved in various metabolic processes such as oxidation, biosynthesis, and long-chain fatty acid beta oxidation. This also contributes to the dynamic nature of this organelle, as between species, tissues, and cell type, the matrix enzyme components can differ greatly. A peripheral region of higher electron density called a marginal plate is also present in some species. This plate is made up of crystalline structure of certain enzymes. There are many stacked flat sheets associated with each other to form the marginal plate. This plate is found in close proximity to the plasma membrane and is closely associated with the membrane. Within the matrix, another region of higher electron density may also be present. This is called a crystalloid core. This is also variable as it is present in rat hepatocytes but not in the hepatocytes of humans. This core is made up of tubules arranged in parallel bundles. These tubules are hollow and the enzymes such as urate and xanthine oxidases are localized to the lumen of these tubules. This is an important factor as it results in the compartmentalization of the peroxisome via enzyme and therefore functional segregation. To contrast, enzymes such as catalase are found only in the peroxisomal matrix and not in the core. Between different species, for example, Saccharomyces cerevisiae, which is a yeast, mammals, plants, and other fungi, there is also great diversity as to what peroxisomal proteins, called peroxins, are expressed. I will come back to this later, but to portray the interspecies diversity, I've used this example. In most fungi, PEX20P and 7P recognise PTS2, but in yeast, PEX18, 7 and 21P perform the same role. Comparably, in mammals and plants, PEX7P carries out this function. In every peroxisome, complex protein import systems are present, for example for long-chain or branched fatty acids. Other common features of all peroxisomes are organelle division machinery and ROS, being reactive oxygen species, metabolic machinery. Catalase is one of the most common peroxisomal enzymes, and as previously mentioned, an oxidase crystalloid core may or may not also be present. Here we have a scanning electron micrograph of a peroxisome, and note its close proximity to the mitochondrion and smooth endoplasmic reticulum. If I lift up the perspex, you should be able to trace the region of higher electron density which I've shaded on the plastic. This represents the crystalloid core. Similarly, in this scanning electron micrograph, we can clearly see the labeled urate oxidase is localized to the crystalloid core, whereas catalase, in this scanning electron micrograph on the left, is present throughout the whole peroxisomal matrix. One key point to note about this organelle is that it contains no ribosomes or DNA. Therefore, every protein which is required by the peroxisome is synthesized in the cytosol and imported in. In yeast, this is the organelle with the highest protein concentration, and the proteins within the peroxisome are heterogeneously distributed. Peroxisomes are also able to import pre-folded and oligomerized proteins. In order to perform these tasks, peroxisomes require complex protein import systems. To date, around 32 known peroxin proteins, or peroxins, have been identified in humans. The PEX genes encode the peroxins, and around 23 PEX genes have been isolated and identified, which are essential for peroxisome assembly. Here is an overview of the organization of some of the peroxins on the peroxisomal plasma membrane. PEX5P and 7P, which were previously mentioned, are free in the cytosol. These peroxins recognize specific PTS signals which target proteins for peroxisomal import. PEX5P recognises PTS1, which is a C-terminal peptide, and PEX7P recognises PTS2, which is an N-terminal nonapeptide. PEX17P, 14P, and 13P are embedded within the peroxisomal membrane. These three peroxins form the peroxisomal docking complex. PEX8P acts as a connection bridge between PEX13P and PEX12P. PEX12P, PEX10P, and PEX2P form a ring complex in the plasma membrane. These are ubiquitin ligases causing ubiquitination after protein import. They are all zinc-binding ring finger proteins. These bind to PEX4P and 22P. PEX15P, 6P, and 1P are plasma membrane bound and are responsible for protein export. Here is a list of references and extra reading if you are interested in learning more about peroxisomal structure, function, and biosynthesis.